Okay. Now, there is so much to dig into here, but I wanted to start with a sort of abstract idea, because this is all about memory. And I just wondered if you could tell us of a memory you have from perhaps a long time ago that we can use as an example.
I guess something relevant to our discussion today. We moved to Germany in 2009, and a memory I have from 2008 is when Gilles and I decided we needed to tell our daughters we were moving to Germany. And we met at the breakfast table in Pasadena, California. And we told the girls, who were then 5, 9, and 13, we have some news. We're moving to Germany. And there was a little shock and sort of nodding, and then, well, why? Well, Dad and I are setting up a new brain research institute in Frankfurt. And they said, okay. And then Charlotte, who was 9, said, but wait, why are we moving? And we said, well, we're moving to do our science there. And she said, well, people don't move. And Gilles said, yes, people move. We move to different places. And she said, people move to Seattle. They don't move to Germany. And so, you know, the end of that story is, of course, that the girls really enjoyed the move, and if you ask them if they would do it again wholeheartedly, they would. It's been fabulous for them. But we all laugh about this family memory.
Who would move to Germany? But the lovely thing about this, of course, is that memory is a critical part of being human, and you've just demonstrated it beautifully. You know, we identify ourselves in terms of our memories and those stories, those moments in life, which are really important to us. And the mystery, of course, the scientific mystery, is that it's all in here somewhere. That you know, in the physical nature of a human brain, somewhere there is an idea that you were sitting at a kitchen table and explaining something to a child, and that is an astonishing thing. Just talk to us a bit about that. This incredible idea that little molecules in here can somehow conjure up your kitchen table.
Right. So, I mean, when you think about memory, there's so many components to it, so many sensory systems. You know, your vision, the memory of the conversation, maybe even the memory of the smells of the breakfast table. So all those things come in via their relative sensory channels, the eyes, the nose, the ear. They come into the brain, and of course, the information is coordinated and integrated at the connections that we saw in the video, the synaptic connections. And our current thinking is that the information is stored by altering the strength of the connections between those relevant areas. So the sensory pathways that converge in a certain integrative area, those synapses there are altered, and they're altered for long term. I mean, this is, I think, still the big mystery, and the thing that keeps us going is how you actually get. It's easy to think how to set something up, but how do you get it to persist when the molecules themselves, the proteins, as we heard in the video, are rather short-lived? They live for days, not for years.
Well, there's plenty of science still to do. So let's just spend a while on how you got to the stage of moving to Germany to do brain science, because you didn't start out in neuroscience, and you went through, there's this wonderful transition in your career where you went from working with human twins, identical twins, to sea slugs, yeah. Which is quite, those are two quite different experiences. Just tell us how you go from the twins to the sea slugs.
So, as an undergraduate, I was doing my honors thesis on these twins. I was studying learning, so I was already at that time very fascinated in how the brain is storing information. And as you may know from twin studies, you can infer how much genes and how much environment contribute to cognitive abilities. And so I came to the conclusion that 50% of memory ability is genetic and 50% is from the environment. But the process of doing science, at least for me as an undergrad with humans, was chaotic. I was driving my VW Bug to people's houses to test their twins, and there were too many uncontrolled variables for my liking. And so I think that that's the reason I migrated to a system that arguably at the time was one of the only systems where you could really study learning and get resolution on what the brain was doing, was in these invertebrate species, which show magnificent learning. So the animal that I studied shows Pavlovian conditioning. So it learns to associate a stimulus with a natural stimulus, and you can change the animal's behavior. And I was able to poke electrodes into brain cells and, really study memory, what had changed in the brain as a result of this behavioral experience.
Amazing. And of course, this is the way science goes, that these paths aren't always obvious. And I love that about it, that there's so many things to study. Let's cover a little bit of the basic science, because I think that one thing that we've almost got used to when we talk about biology is cell theory, this idea that the basic unit of life is a cell. And we see that as a small thing. I can't remember the number, but we are made up of billions or trillions of cells. And so we think of a cell as a small thing, but then actually every single cell is a little factory. So just tell us a little bit about a normal cell before we get to the neurons, and this amazing little factory that each little cell is.
Right, so within the cell, as you say, we think of the cell as unitary, but it is full of small little compartments that are doing their own job, right? Little factories that are making proteins, making energy, degrading proteins, getting rid of contaminants. And the thing about the cell as we see it is that it's hard to visualize those little areas within the cell. So the neuron, in some ways, can be viewed as an opportunity to understand the function of the cell, because it has done us the favor of moving those subcellular compartments and factories out and separated them from the cell body so that we can see them better.
So that's what we saw. So a normal cell is more or less a lump, but then the thing that's different about the neurons is they've got these extraordinarily long things that come away from them. And just tell us a little bit about, so the thing that's at the end of all these long tendrils, those are the synapses, the connections. And so tell us a little bit about how, clarify, we heard a little bit in the video about where the proteins were generated, but why was it a big deal that the proteins were generated in these long fingers rather than in the cell body?
Right, so our understanding of how the neuron as a cell worked was based on decades and decades of beautiful work done on these round cells. And so the assumption was, when people first looked at the neuron, was, oh, well, I see the round cell. All of that other stuff is where the synapses are, but I see the round cell, and so I just need to copy-paste my thinking of how that round cell worked right onto the neuronal cell body. And it just was never questioned that all of the machines in the cell body were still in the cell body, and those machines had the heroic task of actually sending all of their products, the energy, the proteins, all of that out into those processes, which I don't know if your eye could allow you to assess the relative volume, but the axons and the dendrites, the processes, are the majority of the volume of the cell. So historically, we were asking a relatively small little ball to supply all the goods that are needed for this massive volume. So in retrospect, it makes sense what we've discovered, but at the time, the thinking had just transferred the cell body from other cells to neurons, and so that was the dominant assumption.
And let's just talk a little bit about how you got there, because, of course, to see the insides of a cell functioning is very difficult because you can't just open it up. You might be able to put an electrode into a sea slug, but you can't do that with a cell, because if you burst it, it stops functioning. So how do you see inside the cell to see how things are happening?
Right. So, I think, like many things in science, we got into this very indirectly. So we were doing experiments, actually recording synapses with an electrode, but very far away from the cell body. So we were way out where the synapses were, and we were studying how synapses change. And we had made the synapses stronger with a drug. And there was a lot of discussion actually dating back from the 1950s, and people had been ruminating on what is the physical substrate of memory. And there was already knowledge that, actually from whole animal experiments, that in order for animals to show memory, the brains of those animals needed new protein. And this was done by injecting inhibitors of protein synthesis into animals during learning.
So just to clarify, if you can't make new proteins, you can't form memory?
Long-term memory.
In the long term.
Yes,
yep.
You cannot form a long-term memory. So we did the sort of in vitro version of that experiment, and we put on a drug that blocks the synthesis of new protein. Now, if the protein is coming from the cell body, you would imagine that when you change the strength of the synapse, that you would have to wait some time for those new proteins to come and be delivered. But instead, we had a very surprising finding that there was an immediate effect of applying that drug on the learning we were looking at. So it told us it was within minutes that we could see a difference when protein synthesis was blocked. So then we went on to do experiments to really physically isolate these synapses and show that the protein synthesis was coming from that local source. But what's interesting in what you suggested, actually, is that this was a very indirect method, right? We have an electrode in a piece of brain, and we have a drug. And I'm trying to convince myself and everybody else that inside these synapses, there are machines and there are messenger RNAs that are making proteins. So that's the rest of the story, really, is doing those kind of experiments.
It struck me when I was thinking about this that it's a bit like supplying the International Space Station, that, you know, people always had to put things on a rocket and everything had to be carried and everything has to be carried there, and it takes a long time. And of course, now people are thinking about 3D printing spare parts on the space station. And it's sort of a similar, it's much quicker, it's obvious, it's much more efficient. But when you first suggested this idea, I mean, it's always difficult when you sort of suggest a new idea in science. What was the reaction when you first said, well, maybe the things we had been thinking, maybe we weren't getting that right?
The reactions were very strong and very discouraging. They went from, that's crazy, I was junior at the time, right? So some senior people tried to gently guide me away. You're not really going to work on that. And then I really decided I really want to study this. And I remember people sort of, you know, raising their eyebrows like, oh, you're going to really invest in that. You're going to go for that. But I hope that maybe I've conveyed how interesting this is as a problem. And I really felt captivated by it as a logistics problem and as a problem of how cells work. And so, and I guess that we weren't deterred by that. In fact, maybe I was in a weird way kind of decided to prove everybody wrong.
There's an element of stubbornness
yes,
in that, which
definitely.
is very, you know, that it's not always a bad thing.
Yes.
And so once you had the idea that this is what's happening, was it obvious what to do next? What kinds of studies to do to take the science forward?
It was also, I should say that, you know, this work was not done in a vacuum. So I think part of the inspiration that we were on the right track came from some contemporary discoveries. So Kelsey Martin working in Eric Kandel's lab and Christine Holt working in a developmental system both had similar discoveries. So in that sense, I also knew I wasn't crazy, but it was very clear. I would say the roadmap was very clear. It was really terra incognita. We knew very little about what the components were out in the dendrites and axons because, of course, everything had been focused on the cell body. And when I say components, when you think about making a protein, you need the template, the messenger RNA, which hopefully we all know about after COVID, saved us, the messenger RNA. You need the machines. So you need the machines, the ribosomes, which are going to read the messenger RNA. And these were huge things that we needed to look into because at the time, the number, let's say the diversity of what could be made. So neurons express, probably individual neurons express 7,000 different proteins, and only 10 messenger RNAs or so had been seen in the dendrites. So this said, if this is an important process, are these 10 messenger RNAs actually going to change the synapse? So that meant we had to discover the messenger RNAs and see what that population was, to discover the potential for what could be made at the synapse.
So the messenger RNAs are kind of carrying the recipe.
Correct.
And then you have all the material. Presumably you need transport networks that are taking the raw materials.
Yes,
so you up and down.
You have to transport everything, right? So things can diffuse in cells, but most of what gets out to the dendrites and axons travels on these superhighways with motors that actually drive them. So that is required. You need, but the thing that's interesting also in terms of a numbers game is that a messenger RNA can give rise to many copies of proteins. So you get a lot more bang for your buck by transporting the messenger RNA, because you can make 100 or more copies of a protein, whereas if you had to transport 100 proteins, that would cost a lot more.
It never ceases to amaze me how all of this intricate machinery, you know, you're talking about individual molecules moving around, these little ratchets that are moving things around the cell, and somehow we can see into that without being able to see it. You know, it's really impressive detective work. Do you feel like a detective?
There are certainly aspects where we do feel, and then there are moments when you actually see things. So I would say that we're at the cusp of actually increasingly being able to really see these things. So the motors that you're describing, we can see now. So we can see the machines moving along the highways and transporting things. And I'm sure everybody who does this kind of work feels the same way, like how amazing it is to actually see the thing that you've been studying and sort of inferring, and, you know, all the pieces of evidence line up and tell you that it's got to be there. And then when you actually see it, it's really amazing.
And humans, we do say in English, at least, seeing is believing.
Yes, yes.
You're definitely sure you're right.
Yes.
Well, just let's talk about the significance, because, of course, it's very interesting. You know, there's lots of humans like solving puzzles. We like to understand things. So it's interesting for its own sake. But once you know this, what happens next? What can we do with that knowledge? Or how does it change how we think about, you know, memory formation in humans? Why is it important?
So there's two things. One is I'm a champion of, I find just understanding incredibly gratifying, and I think it's something that it's a luxury to have as a human, that we can understand the universe and that we can understand how things work. I think we, like other animals, are naturally curious and we want to understand relationships between things. So that's one aspect. And then there's the more also human and practical aspect, which is that, as mentioned in the video, I think over the last 10 to 15 years, there's been a surprising convergence in many fields of pathology, neurodevelopmental disorders like autism spectrum disorders, neuropsychiatric disorders like schizophrenia, and also neurodegenerative disorders, that, of course, we know that these are brain diseases and therefore something is wrong with the brain. And we've measured the electrical activity and we know that the communication has broken down. But there's been really a focus and an appreciation that many of these disorders converge on the synapse. So they're not just, you know, general communication disorders. Many of the proteins, actually, that are the risk factors for autism spectrum disorders, for example, are actually proteins that are present at synapses. And many of interest to us is many of those proteins actually have their messenger RNA and are mostly made locally. So after years of doing this very basic research, we see ourselves now very close to important human clinical questions. And I think we're in a position where we could really understand the core problems at many of these disorders, because we're in a position now where we have the synapse in focus, and we're able to see the mRNAs and the proteins that are there. And then, of course, once you can see the synapse and potentially control the components, you have potential avenues for correcting things with therapies.
So we should be clear that, any potential therapies are coming from this idea are a long way off. This is very basic and it's really important, but it's almost just the first step.
Correct. So in fact, I think it's very important that we all do what we're good at, right? So throughout the course of my career, every time we make a discovery, there are either students or postdocs or collaborators from the outside who want to come and want me to study a particular disease. And I have felt that my energies are best spent at what we do really well. And that is to study these very basic processes and develop new tools to look at them with better resolution. But so for the first time, actually, I feel that we've got enough information and the tools are so good, and we do have models available. So for the very first time, we are now setting up to do experiments where we will look at a few disease models. So these are mice where a model is developed for a specific disorder. So if a disorder, like one of the ones we're going to look at is Fragile X syndrome, which is a single protein that is essentially deleted. And this is a particularly interesting example because this protein is present at synapses, and it actually regulates whether proteins will be synthesized or not. So there's a mouse model that we can look at that has the same genetics as humans, but also has some of the same symptoms as humans, and we will be able to look at these synapses in detail and see what's different about these synapses. We can do the same thing for other diseases too.
Brilliant. Well, what's great about this is you don't sound like you've finished.
Nope.
Well, just on the broader question, I'm interested in, the Körber Science Prize obviously recognizes European science. And I was wondering how you found working in Europe versus working in the USA. Obviously, they have different strengths and weaknesses. And I was wondering how you found that transition.
Yeah, so, I think I've now spent about half of my time as a team leader in the US and in Germany. And one of the things that I've really appreciated about working in Germany is actually probably represented by all the people in this room, that it's in the culture and in the education of people in Germany, and I hope I could say in Europe, that science is important, that we can understand the universe, that facts are important, that there's a logic and a reason, and if we research things, we can understand how life works. So there's a respect that I've felt working here, also as evidenced by the enormous contributions of taxpayers to what we do, that is sadly, I would say, being challenged in the US. And that's, I would say, a really big difference between the two places.
Great. Okay, let's talk a little bit about the future. As we said, you've not finished yet, but also about the structure of science, because I think before we get to what you'll do with the prize money, I know it's very important to you that science is representative, that we know that the science that is done is not objective, right? It does depend on the people who are doing it and on their incentives. And you have put a lot of work into encouraging groups who perhaps haven't traditionally been part of the scientific world to come into science. Could you just talk about why that's so important to you?
Well, I think it's part of maybe what has been articulated recently when we think about diversity. But if you think about the hard problems we have to solve, there are many solutions and there are many approaches, and we all think differently. So I think the more minds we have to approach a problem, the better. So, and given that the way we think is influenced by our genes, but also by our culture and our upbringing, I think the more cultures that we bring to the table to think about how to approach these things, the better. And the more, that includes men and women. So that's my philosophy, more or less, that it really is about solving the problem. There's also the issue of fairness, but I think one need not invoke that, because it's really, if your goal is to solve the problem, you need the best minds with the most different perspectives to come in to think about how to do it.
And of course it takes, yes, that's very important. And of course, you know, these are complex problems, and that involves a team. And I know that some of your team are here in the audience today, and that you're proud of your team. You recognise they're part of this as well.
Yeah, I mean, so I'm sitting here as the winner of this prize, but you know, I have worked with incredibly talented students and postdocs, and also amazing staff. I mean, the people that I invited here today are the technical assistants and my assistant. They are absolute masters of their craft. They are the reason that we can do things like grow those neurons in those chambers. They are incredibly skilled. They have a discovery mindset, and they're really part of our success. And one of the things that I feel so privileged, you know, being a scientist, is how lucky we are to actually do this job, to be able to discover things. It's a real luxury, I would say, as a human. And I think we need to make sure that everybody who's involved in this enterprise feels part of that success, because they're so essential for the success. And so this is something that we've tried to do in our own institute, which is to first show our appreciation, but also let everybody have a little bit of ownership of what we do, because of course these are really hard problems, many very difficult methods, many widgets to make in the machine shop, you know, IT needed, temperature control, all of that. But it's, you know, if you just look at that, you think, oh, you're controlling the temperature of the room and the humidity. But actually, no, if you don't do that right, everything collapses. So it's really, you know, the whole enterprise is so important. And, you know, I feel a great deal of appreciation for the people who I have with me now, and a lot of respect. But also, I would say, over, you know, the decades that I've been doing this. Yeah.
Brilliant. So I think that's really important, partly because it changes the stories we tell about science, because the stories that perhaps you and I were told at school were that, you know, there was a lone scientist and they worked in their lab, and then 20 years later they emerged with a solution to some massive problem. And of course it wasn't really true then, and it's certainly not true now. And I think we need to change these stories. So stories like yours are really important. Now, of course, the Körber Prize comes along with a great big chunk of money, and you get to do lots of interesting things with that. So tell us a little bit about what your plans are for the prize money.
So I mentioned this just a few minutes ago. So what we've decided to do is to take a pipeline that we've been working on. So if you think about brain synapses in general, you think about the brain packed with probably 100 trillion synapses. Those synapses are not all the same. So different brain areas have to have different properties because they carry out different computations. And we know about how those brain areas function in terms of their electrophysiology, but we don't actually know about the synapses, which are really the business end of the thing. So we've developed methods that allow us to isolate certain types of synapses from certain cell types and look at them in isolation. So if you think about it, instead of taking the whole brain and getting all of the synapses, where you might have, you know, black synapses and white synapses, and when you mix them all together, you just see gray. So you would just conclude, and actually there's really no gray, right? There's no gray synapse. There's a black synapse and a white synapse. So it can be incredibly misleading to average, basically. So the beauty of the pipeline we've developed now is that we have this resolution to get synapses from certain cell types. And with that resolution, we're now going to look at these disease models. So we're going to be able to really say it's this cell type in this brain area and that set of synapses that is particularly affected. And along with that comes, of course, if one were to imagine clinicians thinking of a therapy, that resolution is very important, because many of the inroads we have for therapy need to be more specific. You don't want to treat an entire brain when a very small nucleus in the midbrain is really what the problem is. So this kind of specificity and targeting will be really important for therapeutics, I think, as well.
Brilliant. It sounds like there's loads of exciting things to come. So just to finish then, I mean, scientists are good at generating questions. You know, you can be good at answering a question, but you have to ask it first. And I was just wondering, you know, looking ahead, is there one big question that you'd love to get an answer to? It's always hard because there's always too many questions. But is there, if you could, you know, if someone said, you know, you can choose, and in 10 or 20 years, we're going to guarantee you can answer this question. Oh, what's your question?
Wow, that would be a gift. For me, right now, I've spent, you know, the last few years thinking and talking about these ideas, and the one thing that I think we're just about poised to do is to look at the persistence of memory at synapses by following the proteins. So we can take snapshots now, but I would like to actually look at a memory over time and see how it manages to persist in the face of this ongoing turnover of proteins at synapses. And I think we have the tools now where we may be able to do that. That's a dream experiment for me, maybe a bit nerdy, but...
This is the Science Prize. We are allowed to have nerdy dreams. Erin, it's been such a pleasure to talk to you. Thank you so much.
Thank you.
Great.
Sit down.